All right, let me share my screen. Just kidding. Oh my God, we're here for the first time in years. I'm so glad to have you here. Good morning. Welcome to Talking Science 2023. My name is Jeannie Garbarino, and I am the Executive Director of RockEDU Science Outreach here at the Rockefeller University. The last time we were able to hold Talking Science on campus, it was 3 years ago, in January of 2020. And I remember it quite fondly because I'm pretty sure I tried to do the Running Man with a bunch of you and I totally tore my hip. That's what happens when you get older. But just a couple of months later, we all experienced a profound shift in our lives with the onset of COVID-19 pandemic. Like many other events and activities over the past few years, Talking Science was held virtually in 2021 and 2022. And so it is just so cool to be back with you all in person in Caspary Auditorium to hold and introduce today's event. P.S. There's been a lot of really famous people who have stood on this stage. And so I encourage you to take a look at some of the archived videos that we have online. One of the coolest people that has ever, I think, that has been on the stage is His Holiness the Dalai Lama. Can you believe that? That's so cool. So yes, okay, this is not post-pandemic, I shall not say that, because we are not out of the pandemic, and your safety is of the utmost importance to us. And as you know, personal circumstances and comfort levels vary greatly from person to person, and we try to be as inclusive of a space as possible. And so with this in mind, we have 2 key requirements for your participation today, and this is very important. First, each of you have received a KN95 mask. As you entered the building, they have our little cool logo on it, so they're not super boring. So please keep this mask on, or maybe one that you've brought yourself, covering your nose and mouth at all times. The only exception to this rule is when you're eating or drinking during lunch. Secondly, please do your best to maintain as much social distance between yourselves and your fellow attendees as possible. Try to spread out if it looks like a crowd is forming, things like that. So everybody cool? Yeah, it's the norms, our new norms, I should say. So today we are going to devote this morning to exploring the brain and memory with Rockefeller neuroscientist Dr. Robert Darnell, who will be talking about riveting memories, how they are made and lost. But before I introduce Dr. Darnell, I want to tell you a little bit about Rockefeller University's history. More than 120 years ago, the institution's founder, John D. Rockefeller, recognized that a major barrier to preventing and treating diseases is ignorance about their fundamental causes. So in 1901, he created the Rockefeller Institute for Medical Research in order to bridge this gap. In 1955, there will not be a test, by the way. Just enjoy the facts. In 1955, the Rockefeller Institute expanded its mission to include doctoral education. So here, while we are a university, we don't have undergraduates, but we have Ph.D. students and M.D. Ph.D. students. And that's when it became, 10 years after 1955, so 1965, because I can do math, that's when it became the Rockefeller University. So there are approximately 200 graduate students on our campus each year, working along with 600 scientists who range from postdocs. So postdocs are folks who get a graduate education, so they get their PhD, and they decide that they want to do a little bit more and really learn a really strong focus, and so they become a postdoctoral researcher. And we also have distinguished professors who head the university's more than 70 laboratories. Our mission has long been science for the benefit of humanity, and Rockefeller scientists conduct basic research and clinical research, and their investigations touch on almost every disease or health condition that you can think of. Some of the many accomplishments and breakthroughs of Rockefeller scientists are listed on the back of your program, and they also include the landmark finding that DNA is the basic material of heredity. This happened in 1944, and just as a note, the Nobel Prize for the structure of DNA was not given out till 1954, so they needed this information in order to make that happen. The discovery of the hormone leptin, which regulates appetite and energy metabolism. The discovery of the immune system's dendritic cells, which are crucial for fighting infectious agents and cancer, actually. And the development of a combination therapy that transformed HIV infection from a fatal disease to one with a normal lifespan. Over the years, the importance of the discoveries made at Rockefeller has been recognized throughout the world. In fact, 26 Rockefeller scientists have been awarded the Nobel Prize, which is the top prize in science and medicine, including 2 who have received their awards in the last six years. In 2017, geneticist Michael Young was awarded the Nobel Prize in Medicine for his discoveries about the molecular mechanisms of circadian rhythm, the process by which our sleep and wake cycle and metabolism are regulated and coordinated with the Earth's 24-hour day-night cycle. And in 2020, virologist Charles Rice received a Nobel in Medicine for his research that made possible the development of a cure, an actual cure for hepatitis C. Let me tell you, coming to work every day is nothing short of extraordinary. This place is amazing, and I'm so thrilled that you all get to be a part of this. But this university does more than just conduct great science. We believe that Rockefeller's mission also includes sharing our science with students and teachers. In fact, RockEDU Science Outreach works year-round to provide fun, authentic science experiences to thousands across the city. Some of our events include informal gatherings and mentored research programs, such as Lab Jumpstart and the Summer Science Research Program. We have a couple of students in the audience from those programs, and I hope that we have a couple of students who are joining us for 2023 summer. And you can find out more about these programs and other science activities on our RockEDU website, rockefeller.edu/outreach. And we hope that you'll come back with some of our other RockEDU programs. All right, let's get to today. So today we are pleased to welcome students from more than 50 schools. That's so exciting. From across the 5 boroughs of New York and beyond. Thanks so much again for spending your Saturday with us in pursuit of scientific knowledge. And I also want to just give a quick set of snaps for your teachers. This is a Saturday. They did not have to come and take you here, and yet here you are. Thank you to the teachers for being at the front line of science education. RockEDU and Rockefeller University values the work that you do every day. We are also deeply grateful to the Andreas C. Dracopoulos Family Science and Society Initiative and Bristol Myers Squibb for providing their generous support that enables us to present this year's exciting program. Okay, now all of my boring stuff is over, and I get to introduce the star of the show. It is my pleasure to introduce the amazing Dr. Robert Darnell. Bob, we call him Bob. It's like one time I went to a meeting and I saw Robert De Niro, and then I realized all of the celebrities also call him Bob. So you're like a celebrity, Bob, and we're just going to call you Bob. Bob is the university's Robert and Harriet Hellebrand Professor, head of the Laboratory of Molecular Neuro-oncology, and a senior attending physician at Rockefeller's Research Hospital. A renowned neuroscientist and neuro-oncologist, he works at the intersection of immunology, neurology, and cancer biology. That's pretty cool. He is also a leader in translational research that turns fundamental discoveries into new medical advances. Some of Dr. Darnell's earliest research focused on neurologic disorders, or paraneoplastic neurologic disorders, or PNDs, which are a group of devastating neurological conditions that occur sometimes when the body reacts to the presence of a tumor. Dr. Darnell's study led to the discovery of neuron-specific systems that regulate RNA in the brain. In the course of this research, he developed new methods called cross-linking immunoprecipitation, or CLIP, to understand the role that these systems play in neurons and other cell types. Bob came to Rockefeller in 1992 as an assistant professor. Since 2002, he has also served as an investigator with the Howard Hughes Medical Institute. With a lifelong commitment to translational science, Bob helped Rockefeller weather the storm of COVID-19. By adapting a genetic testing procedure created as part of his lab's work on RNA biology and inflammatory diseases, he developed a simple, safe, and self-administered PCR saliva test to detect the virus. Over the past few years, his lab has processed nearly 200,000 tests at a rate of about 1,500 tests per week. So everybody, like everybody at Rockefeller, we all just spit in a cup and put it in a tube and put it into the drop box, and we all every week we know if we're positive or not positive, and it's just been such an incredible force to ensure that everybody who comes to work is doing so safely. It's just an amazing technology. And I appreciate it as a person who has to take the test on a weekly basis because we're not getting a swab up our nose. So that was really cool. So Bob received his undergraduate degree from Columbia University and went on to earn his MD and Ph.D. degrees from Washington University in St. Louis. Bob has received numerous awards, including the National Institutes of Health's Outstanding Researcher Award and the NIH Director's Transformative Research Award. He has been elected to the National Academy of Sciences and the National Academy of Medicine, and he is a fellow of the American Association for the Advancement of Science. In today's program, Dr. Darnell is going to talk about how his recent cutting-edge research in molecular biology and genetics is revealing mechanisms that underlie memory and learning. He will also discuss how these processes go awry in conditions such as Fragile X syndrome, autism, and Alzheimer's disease, and how this knowledge can be used to create better therapies. You have plenty of time to ask Dr. Darnell questions following his talk. Be sure to, we gave you guys some really cool snazzy notebooks. Those are for you to jot down some notes. Maybe you have a question that you want to save and ask for the Q&A portion. So those are really helpful because during the Q&A portion, we do have special prizes if you're able to ask a question. So it'll be very exciting. At noon, we will go outside and across the lawn. Some of you have already been into the CRC building, but if you don't, we have people that can show you where to go. And we'll have lunch there and a bunch of interactive activities. You'll also have the opportunity to interact with some current college students, some current graduate students, postdocs, and also some professionals in the STEM space so that you can learn about what it's like to figure out a career and education in science and learn the diversity of ways that that can happen. You'll also be able to hang out with Bob at the Ask Bob Anything mini talks in Caspary Auditorium. And with that, I am going to welcome Bob to the stage, who will start his amazing talk on Riveting Memories. So thank you, Bob, and welcome.
Wow, oh, that was a long introduction, but thank you, Jeannie. Jeannie's an amazing asset we have here at Rockefeller, and she preempted some of the things I wanted to say, but first and foremost, I want to shout out to all of you guys for waking up on Saturday morning on a long 3-day weekend and getting yourselves here. So all of the high school students, I'm proud of you for doing it, and also all the teachers, I'm so grateful for you both being here and encouraging your students. And I just thought, for 1 second, can I take a picture of you? So I want everybody to smile, but you have masks on, so it's not going to show, but you know, what the heck. Yeah, that's so cool. All right, we'll post that somewhere. All right. So I'm going to talk about memory, because memory is, in essence, one of the things that we do that distinguish us from each other and from other species, and yet there are many common features. Animals have lots of memory, that's really important, but the way we put memories together underlies the way we have consciousness and the way we think forward into the future for our planning, for what we want to do when we grow up, which I'm still trying to figure out. How do memories get put together? Now, one instance of this that I like that was illustrated in a book by called Gödel Escher by Hofstadter is this idea of coincidence. So one of the things, like I've heard many, many lectures in my day, one of the things I've come to realize is coincidence of what I had to say, plus the sound of my voice, plus moving across the stage, which really good speakers do, and maybe some attention-grabbing things coalesce to help form a better memory. And so that's illustrated here. This is something we all sort of have experienced, right? A memory of something that you haven't thought about for years. Since you were a little kid, 5 years old, 6 years old, playing on the beach, for example, or doing something in the city that had different modalities to it, like movement or sound of the ocean, or the smell of the waves, or hearing birds in the background. And something 10 years later, or longer in my case, could be 50 years later, all of a sudden will remind me of that, because I'll be on a beach and I'll hear a sound and I'll hear those same birds, and it'll be in the part of my vision that I was paying attention to, and it'll all like instantly jump into my mind. Something I hadn't thought about maybe for many, many years. So this idea of coincidence detection and as an underlying cause of memory is something I will come back to over the course of this talk. So what does this sort of boil down to? It boils down to what is going on in your brain to make memories. So the brain, I'm just going to briefly remind you, has different features to it. This is an actual human brain. So for scale, those are two hands. It fits inside your skull, fortunately. And it has what we would generally call the cortex, which is most of these gyri that have little, what we call sulci in between them, separating out different areas of the brain. And we artificially divide them into regions like the frontal lobe, the parietal lobe, the temporal lobe, and so on. So this is the main area that we use to form and store memories, and also to coalesce memories into plans and thinking about the future. There's another area of the brain that's a little bit separate. It looks different here, called the cerebellum, and that's involved in motor control. And motor memories, I'm going to tell you, we all know, we all have them, but I'm going to think through it a little bit with you, what they are and how they're distinct from memories we have in the cortex. So there's teaching points that we can extract from these two different kinds of memories. I'm calling them here conscious memories and unconscious memories. And then this, just to orient you, is an MRI scan cut right through the middle of someone. There's no actual cutting, right? But you can see the cortices here, cerebral cortex, and you can see the cerebellum there. And this is the brainstem area I'm not going to talk about, although it's super fun. So within the brain, of course, are individual cells, and we call these neurons. These were recognized many, many years ago and drawn very beautifully by a neuroscientist, Ramon y Cajal. And you can see he literally drew pictures of individual neurons, which are these black dots here, but each neuron sends out branches. So they're very different from a red blood cell in your blood or a lymphocyte. Neurons are unique, and of course I have neurons in my brain in the cortex, the motor part of the cortex, that on command I can say, wiggle your left foot, right? So those neurons have to basically go down to the spinal cord many, many feet, and then a second neuron goes and wiggles my foot. So these extensions can be very, very long. And for each neuron, it's worth noting, of course, this is ultimately mediated by instant or semi-instant electrical signals, right? That I can ask you all to wiggle a finger now. I got some wiggles, right? So but that happens more or less instantly, right? So your brain is hearing me in the auditory cortex. It's sending a message to your motor cortex, and instantly you can fire and wiggle your finger. So this process is something we want to think about in terms of understanding not only an auditory instant command like that, but how a memory gets laid down for this lecture. And one of the interesting points about this is looking in more detail at an individual neuron. This is called a Purkinje neuron. It's one of the main neurons in the cerebellum. And it has many, many processes that are more or less within reach of the cell body, within millimeters. And then it has one output, which is an axon. And that output can go a long way. It's worth understanding here that the neuron itself has incoming receivers, that's the dendrites, and an outgoing firing axon. And fascinatingly, all of the input is integrated, and put a little math symbol up there for STEM crowd, right? All of the input for connections that are made coming into this, these sort of blue electrical wires, are actually integrated in the cell body to make a decision, but this is a mechanical electrical decision, to fire or not fire an axon. So interestingly, for those of you interested in computers and the brain, which is everybody, I will define that for you, is that the axon is really a digital output. It's a 1 or a 0. So the integration of all of this information in one neuron is summed, and the neuron will either fire or not fire. So that has a nice analogy to computer bit. Now, how many of these neurons are there, and how does this integration work? We're going to talk about that. One way to think about that is to look inside a brain. So the technology is spectacularly beautiful now, and will continue to get better over the course of the next 20, 30 years. So you guys have an amazing future in front of you for thinking about the brain. And I'm recruiting all of you to become neuroscientists here, okay? So this is a current picture from an HHMI group at a place called Genelia Farms of a mouse brain where they're looking at individual neurons colored, and you can see sort of the processes like this. Here's another neuron, the process is green, and you can see they're stretching all over the course of the brain. And what you get at the end is either via your interpretation, a real mess or something really fascinating. So in fact, the mouse brain is put together so pretty much each mouse operates more similarly than different from every other mouse. So there's a genetic rule of how all of this gets laid down and allows a mouse to know how to nurse the moment it's born, for example. So what about humans? Humans are a little bit more complicated than a mouse. We have approximately, estimates are about 100 billion neurons, but it's more complicated than that. I won't excite you, not depress you about this. So here's a neuron here. You can see the large cell, the pyramidal neuron. It has many dendrites coming out, and these dendrites interact with incoming axons to make points of connection, which we call synapses. Those are the spots where electricity is transferred. Now, within a single dendrite here, this sort of mess here, each dendrite has little points, little pockets of outreach, which are called dendritic spines, and those are actually the spots where axons come and attach here. And when you start to think about the math here, there's thousands of dendrites in any one neuron per that picture I showed you of the Purkinje cell. And each dendrite has many, many, many thousands of spines on it. So there's a lot of information that the neuron needs to integrate to make this go, no-go decision for the axon. So it's a little bit like a circuit board with, if you do the math, I would estimate about 10 to the 19th connections. So you've got a lot of room to spare in terms of the things you can dream of, things you can think of, things you can learn, and things you can plan for your future. And to zoom in a little bit more, since there's white space on this screen, that's not a coincidence. So here's a close-up of an axon that has decided to fire in the synapse to activate one dendrite on one dendritic spine. And we'll talk a little bit about this, but we don't have time to go into all of the details. But suffice it to say that the axon, when it gets an electrical signal to fire, releases a biologic material. That biologic material are little vesicles drawn in yellow here, like little packets. And inside each packet is what we call a neurotransmitter. The most common excitatory neurotransmitter is an amino acid, glutamate. and they're inhibitory neurotransmitters that can either activate or turn down this dendritic spine, and they work by binding to receptors on the surface of the dendrite. So the glutamate gets released, can bind that receptor, and that opens up the receptor channel for electrical circuit to go through, which is conducted by ions, potassium ion and calcium ions. So that's an overview of the individual microcircuitry. What about stepping back for a minute for a macro view of how we think about this? So this is, again, a picture of the technology. And I put this up here for you guys because Jeannie has set up something for you to play with after class. And Daniel O, one of my graduate students who's sitting here, yay Daniel, as well as another graduate student, Simon's son, and a postdoctoral fellow, Ruth. I'm not sure if they're here. They may still be sleeping, but that's okay. They'll be here in time. To take these amazing things that Jeannie actually found this lovely website where you can rotate live this image. You can see the eyes here, and it's imaging is the different neurons inside of a brain. And you can rotate this, but you can also slice it, like for an MRI scan, to get a picture of, here's a slice through the head, virtual slice, showing the circuitry inside this part of the brain, and here's a slice from a different angle showing this kind of circuitry. So all of this comes together to say these are macro ways that we can image what's going on in the brain. But to understand the memory formation, we need a conjunction of this sort of macro view of what's happening and the micro view of what's happening in the synapse that we went through on the last slide. So it becomes a challenging problem, especially if one is interested in human disease, intellectual function, intellectual disability, long-term memory or failure of long-term memory, as in Alzheimer's. And while I'll talk about different approaches to that, but it's worth thinking of the approach as a human one. So let's talk first about memories in the cerebellum. So these, as I mentioned, I think are distinct memories. I'm going to illustrate to you why I think that and why the cerebellum is important in this kind of memory. So this is something that many of you have done, maybe not all of you, but learned to ride a bicycle. And I don't know if you remember it. I kind of remember it. And it was a few years ago, but it's not so easy when you're learning. This girl looks pretty good, but not sure she's going to fall or not. This dude should be wearing a helmet because you can not only scrape your butt or scrape your knees, but you can scrape your head. So bicycle helmets, and I'm a neurologist, are important for everyone. But if you asked someone, if I asked you right now, conceptually, how do you balance on a bicycle? Can you tell me how? Because I can't. This is one of the things my neurology mentor, Fred Plum, taught me, is motor memory is not something you can consciously articulate. Like exactly how you're balancing on that bike, it's not part of your cerebral cortex, which is where conscious memory lies. In fact, instead, it's part of your cerebellum, where unconscious memory lies specifically for motor movements. So I'm going to illustrate 3 different examples, which I like, so hopefully you'll like them too, of the pure motor memories of this sort that are basically unconscious. And then the way with memorized motor movements, like riding a bicycle, you can ride a bicycle and think at the same time, oh, that's a beautiful mountain. So let me illustrate what I'm talking about with some fun videos. So this is the greatest of all time, Simone Biles. And I did not, this is a movie here, okay, so, and I apologize for not providing popcorn, but there's no eating in this room. I did think about it, Jeannie. But look at her concentration here. So you can imagine how much concentration she has in her, and yet, at the same time, I would bet that she could not tell you exactly how she's going to move her feet and do her twisting. It's a memorized thing that is unconscious. So here's an example just of why I think that, and I'll reinforce this later, but what is going on in her mind right now? Look at her smile. All right, that's freaking unbelievable. Sorry, right? She's not thinking during that. That is like a 4-second clip. She's not thinking about what she's doing. It is memorized, okay? Here's another one, another lovely goat in soccer. So Messi is here, that's his hand. And Messi is different from Simone Biles in that it's not a solo sport. So more like the bicycle and looking around. Messi is got an unconscious, incredible ability to play soccer, but he also has to be, and part of the reason he's great is he has to be conscious of everything around him, every other player on the field. And the greats in sports, in team sports, do this, right? Now you guys know because a lot of you play sports. So here's a nice clip of Messi where, see this guy's legs? Messi's dribbling the ball here, but watch what he's going to do. Boom, right through the legs. And then same thing, his feet are moving. He's not really thinking about that. He's thinking about boom. There's 6 guys around him. So he is aware of his environment consciously. At the same time, he's unconsciously like doing his stuff with his feet. So it's an interesting illustration for me. And the next one is even more so in terms of the separation of motor consciousness and cerebral consciousness. So I don't know how many of you guys play an instrument. Me, I do. I play cello. Lots and lots of hands there, right? So one of the things I know, having been beaten by my teachers when I was a kid, to memorize the music, memorize the music, memorize the music. And really what they're getting at, I think my teachers didn't necessarily recognize it from a neuroscientist's point of view, but the motor movement of playing the piano, once it gets memorized, is stored in your cerebellum. And if you memorize it, what does that free you up to do during your music? Emotion, cognition, thinking about interpretation. What does the music mean to you? You know, this part I really love, right? So look at this video. This is the goat of piano playing, a guy named Glenn Gould, who is believed to be the greatest of all time with respect to interpreting Bach. And Bach, of course, was part of the Gödel Escher Bach book I mentioned. It's kind of Bach intersects STEM thinking in ways I won't have time to go into here. But listen to this and watch what happens to Glenn Gould. So this is him practicing in his home, and one time and one time only. He's a very insular person. He allowed someone to just listen to, film him while he's practicing. He's not thinking about these fingers. Forget it. He's thinking consciously. Boom, his fingers go right back. He doesn't have to think about what his fingers are doing. He's thinking about his interpretation of the gosh darn music. He loves and is deeply integrated with the contrapuntal melodies of what's going on in Bach. That's cortical, that's conscious. And when he sits right back down there, I just love that, boom, his fingers just go, right? Total separation of the cerebellum in Glenn Gould from his cortex. So this is the best illustrations I can come up to explain to you unconscious memory and conscious memory. And how do I stand up here so arrogantly to say that? One of the ways that we learn in complex neuroscience, things like memory that are very, very, very difficult to understand otherwise, is to be a doctor, okay, or to understand clinical medicine. So with respect to the cerebellum, there have been patients who've had to have part of their cerebellum, the cerebellar hemispheres, removed for tumors or other reasons. And when those are removed, patients are fine. They recover from the surgery. They're completely normal. They can walk. They can do, their eye movements are okay, but they lose the ability for complex motor memories. And so, for example, an expert pianist who has his cerebellar hemisphere removed will no longer be able to play piano, but they'll be fine otherwise. And their consciousness, their memories of other things are okay. So clinical teaching is a critical aspect of understanding complex phenomenon in people, and in the context of neuroscience, that means integrating clinical understanding with the molecules in the synapse of the axon dendrite electrical connections. So that's sort of the direction we're going in here. So again, this is a complex diagram, again, of the cerebellum, how these cells are connected to each other. And just to focus here on the thing I showed you before, the Purkinje neurons actually receive, via their dendrites, axonal firing input from these other cells called the granule cells. And the granule cells, what they do, it's just a lovely piece of biology, is they send their axons out, and the axons split and branch parallel to the dendrites of the Purkinje neurons. So basically, you can imagine there are many, oops, well, something bad happened, but that's okay. There's many Purkinje neurons in the cerebellum aligned like this, and the granule cell axons come up like telephone wires and integrate with all of the, or with a large number of Purkinje neurons. So this sort of telephone wire-like, I think I know how to fix that, yep. This telephone wire-like axonal dendritic connection is a lovely illustration and point of science to explore. So what is some of the ways that we think about this as scientists, from this very holistic, clinical, observe the patient point of view down to the molecules? So just to review, the basic molecules you're familiar with, here's a crude diagram of a cell. This could be the cell body of a neuron. There's DNA in every cell that's the same in every cell in your body. But what's different in every cell in your body and different at every moment of every cell in your body is the way that different DNA regions are turned on or turned off, okay? And that is the process of transcription of DNA into RNA. And RNA, as you know, is made as a direct copy of DNA, but it's longer than it ends up being. It has introns that get spliced out to make a mature messenger RNA, and that codes for a protein. The mRNA is transported to the cytoplasm and gets translated via the ribosomes and tRNAs into a protein. So at the end of the day, neurons can make different sets of proteins, and in fact, this is a picture, actually, someone was asking about X-ray crystallography. We go down all the way to the level of looking at a patient and what happens to them after an illness, all the way down to the level of X-ray crystallography. And this is a protein we discovered in our lab called NOVA that is a RNA binding protein that is binding in purple here to a piece of RNA. So the brain, as I'll reiterate, has its own special system for regulating this step, transcription of DNA into RNA and RNA into protein. So RNA I'm going to perseverate on a bit because I love RNA. Now, how do we think about this? How do we come to these kinds of observations? One way to do this in the basic science world is to take model systems. And Rockefeller University has amazing science here. Corey Bargman, Shai Shaham, others have studied this little worm, C. elegans, because in part you can take a fertilized oocyte for the worm and follow every cell that is generated in the entire body of the worm, every neuron, every gut cell, every skin cell, and map it out. So this is a beautiful illustration of the nervous system of C. elegans. So it's a model system for studying nervous system development. Here's a more complicated one studied by Mike Young, who won the Nobel Prize for circadian rhythm, Leslie Vachon and others here, from the fruit fly. And this, you can see, is more complicated than the worm, but again, with molecular biology tools, you can get a very detailed picture of what's going on here. A step further is what we call translational science, which includes animal models. And mice are the most commonly used one here at Rockefeller and elsewhere, in part because they breed fast, not as fast as a fruit fly does, but within 3 weeks, a mouse will go from birth to being able to be an adult and generate a new breed of mice. So you can do a lot of work with mice, and they're mammals like we are. So they're a lot closer to humans than the worm, hopefully. And I just put a metric up here of how fast they develop. 4 days in a mouse lifetime is about what it takes to graduate high school, normalized to the average human. I haven't really thought about that before, but anyway. So the problem is, of course, mice aren't humans, much less a fruit fly or a worm. So while we can learn an enormous amount from these model systems, and each one I could spend a whole lecture on, the bottom line as a neuroscientist who's interested in people and human disease is that we ultimately need to think about people. And we know, as science has progressed in the last few years, mice are both a wonderful model and they fail at certain things in terms of their molecular biology and what they can do. They can't do some of the things we do. So I'm an advocate of at least attending to, and Priya Rajasuthapi here is a junior faculty who just gave a lovely neuroscience lecture where she studies the mouse in great detail, but she's very aware of cortical diseases in humans and applies that thinking to her mouse studies. So, and this is sort of a pseudo quote from a great clinician, William Osler, who said, if we listen to our patients, we can hear the answers. And I should point out, there's a little pearl here. So in clinical medicine, we talk about dropping pearls of wisdom to our students who are medical students or junior doctors. And I attend as a physician in the hospitals around here, so I'd like to drop pearls. And I would encourage you to watch out for pearls in this talk. I forgot to mention that in the beginning, but I put a few pearls in there as key points. So you may have seen, I can't remember if I had any in those first few slides, but this is a pearl in my view, a pearl of how to think about your science. So turning to that concept here, what are some of the clinical pearls that could help us think about cortical function as opposed to cerebellar function, right? So this is much more difficult, but there are instances where we can learn a lot. So here's the brain again, the picture of the brain, and this is a rare and unfortunate, but not unfortunately unique individual who had a brain problem that led to a life change in him. So this man, Phineas Gage, born 1823, died 1860, was a church-going man, father, you know, had a wonderful family, had a job that he'd had for many years, held down as a railroad worker, and just like a stable, what we would consider in some sense a stable citizen. And while he was working on the railroad, he was banging they had these big railroad ties, and banging them, they have a little explosive to help it sort of burst through the rocks to make these big holes in the ground. And this one, unfortunately, and he's actually holding the railroad tie, so he survived this incident, but he banged this thing and the explosion went backwards and knocked it right through his mouth, right through his brain. So this is a reconstruction of the railroad tie, went through his roof of his mouth, right through his brain. And you can see his eyes closed on the same side here. So what happened to Phineas Gage is he had a terrible accident that affected only his frontal lobe. He recovered from the accident, he was fine, but he quit his job. He stopped going to church. He started drinking at the bar stealing money. He became what we would call dissolute in neurology. And in some sense, that the inverse of normal Finney's gauge and Finney's gauge after the frontal lobe accident tells us something about what the frontal lobe does. The frontal lobe is critical for each one of you in planning your future. That's the essence of frontal lobe function, okay? And that planning of the future comes out in thinking, learning, but integrating all the aspects of your life, including this lecture, which will hopefully be part of an integration for planning what you want to do in college. You've already made plans for what school you chose to go to, and you're going to make plans for college, and you're going to make plans for post-college. So the frontal lobe is critical for that. And I would actually just say, I'm going to, off the record, but I wrote the record so I can do this, proselytize. A lot of cannabinoid receptors are located here. We don't actually understand what they do, but Nora Volkow, who runs the National Institute of Mental Health, and I have talked about this, as cannabinoid receptors are part of the things that affect these synapses in ways that are fascinating we could talk about. But it is a little red flag for me with the legalization of cannabis, what effect that may have on long-term frontal lobe planning. So just a little proselytizing as a dad, sorry. So here's another patient. This patient is called patient HM, died only a few years ago. One of the most studied neurology patients in the world. And this patient had an accident not in his frontal lobes, but a doctor-induced damage to his temporal lobes. So what happened in patient HM is he had intractable epilepsy. And to this day, one of the treatments for like epilepsy you just cannot control oftentimes comes from the temporal lobes and can spread from one temporal lobe on your left side, if that's the source of your epilepsy, via connections to the temporal lobe on your right side. So epilepsy oftentimes is bifocal on both sides of the brain and can lead to generalized seizures and loss of consciousness. But in the short term, temporal lobe epilepsy, we could talk about. It's absolutely fascinating, the ways it can manifest. But unfortunately, in patient M and some other patients, that epilepsy can be intractable. Like it can be so disruptive you can't do anything in life, right? You can have seizure after seizure, day after day. So one of the treatments in 2023, like Yale has pioneered this, Yale Medical School, one of the great medical schools in the country, is to take out the temporal lobe as a last resort. If there's a focus in there that's, and epilepsy I should say, I'm sorry, is an electrical short circuit of these synapses from something wrong, many, many causes of this. It's not uncommon, but to have severe epilepsy is extremely uncommon. But Yale can take out a piece of the temporal lobe and people are fine and can cure their epilepsy. They do very, very well. But in these days, it was the early effort to do this. Patient HM, they said, okay, well, this guy's got epilepsy in his left temporal lobe, but it's traveling over to his right temporal lobe, so let's take them both out. When they did that, he woke up from surgery, he was fine. Again, rehab, got out of the hospital, went home, and lost the ability to make any new memories. So his memory essentially was frozen in time from the day before he went to surgery. He could remember his past, his childhood, his friends, what he had done his whole life, but he couldn't make any new memories. So that tells you, again, something important about bilateral temporal lobe function. If you have both of them out of commission, you've got a big-time cortical memory problem. Okay, I'm going to talk more about this. The net of this is we can use these clinical insights to start to gain some traction on these very complicated questions, and they can point us to where we may want to start thinking about molecules. Now these observations are fascinating, but they don't really point us towards molecules, they point us towards macro regions. So how can we make that pivot here? So again, I'm going to show you again this concept that there is a way to do it. And I think that one of the interesting things to me as a clinician and a basic scientist is that this entire pathway is doable. So partly I proselytize for people who are interested in being a doctor to also think there's a parallel way to be a doctor, which is to be a doctor scientist, okay? And that works for me because I love what I do. And I care so much about people, and yet I focused on molecules and animal models in between. So how did I become, I was finished college, started thinking about with my frontal lobes, but I didn't realize it, what do I want to do? And I started thinking about diseases that had been discovered where a single gene went defective. So thinking about genetics, which is very powerful, right? It's reproducible. Everybody who has this mutation will have the same problem. And thinking about what kinds of diseases could there be where people had not just brain disease, which I was interested in, but also, and I learned in medical school, there are some diseases that also had, those patients would have cancer. And cancer pathways were just being discovered when I was in graduate school as being very important for controlling critical things of how a cell works. And yet here were a subset of people with diseases you may have heard of, tuberous sclerosis, von Hippel-Lindau disease, ataxia telangiectasia. There's a whole list called the phacomatoses of people who with one defect can have predisposition for cancer and a predisposition for brain problems. So I started thinking, okay, I would maybe develop a laboratory to try and clone one of those genes. The one I became interested in was tuberous sclerosis. The only thing I didn't like about it was the methodology, which is it's a 10-year project, 15-year project to just put together kindreds of people who have tuberous sclerosis to try and do the genetics on them. And I wanted to get my hands wet in the laboratory. I like doing benchtop science, as Daniel knows. So I came across, by luck, in the course of my training, across a very rare group of diseases that Jeannie mentioned. These are called paraneoplastic neurologic disorders. These define esoterica, like most doctors haven't heard of them. So you guys are in the elite now to have even heard of these things. So here's what happens in these disorders. It fit this sort of paradigm in my head. These are patients who have cancer, common cancers, breast cancer, ovarian cancer, lung cancer, lymphoma, and so on. And what we believe happens is they have a silent process going on in which their cancer cells turn on proteins, this red star here, that are normally only made in the brain, in neurons. And that does two things. One is, because there's a blood-brain barrier, this bar here, the immune system, which is always looking for new viruses and bacteria and stuff like that, doesn't really get into the brain in the way it does the rest of the body. The brain is an immune-privileged spot for lots of reasons. So this immune response out in the body at age 50, 60, 40, in some people can recognize their cancer when it's expressing on brain neuron-specific protein and kill it. So these patients actually don't know that they have cancer. They have what we would call naturally occurring tumor immunity, something that was disparaged, and myself included, nobody really believed in for a long time. But, so they don't come to a doctor's attention. And you can imagine, if you have a successful immune response against cancer, nobody would ever know it, right? I mean, you only know about disease when patients present to a doctor, and then you can work on that pathway from clinical medicine to translational to molecules. But if you have something that's working, but you want to know about, it's very essentially impossible logically to even find it. So the reason these diseases are so fascinating is something else happens, a second event. So this tumor suppression may be happening for months or years in these patients. And then something else comes along, which is somehow, we don't understand what it is, the immune system breaks into the blood-brain barrier, gets into the brain, and starts to say, I found more. I mean, the immune system's not smart. I mean, it's very smart, but it's thinking it's found a virally infected cell, for example. It gets into the brain and says, I found more. Let's attack that too. I mean, when you get the flu, we're I'm not going to talk about SARS, although that's a whole other fun topic. But when you get the flu, it infects your respiratory epithelium, and you get an immune response that kills every flu-infected cell in your body till it's 100% gone. And then you cough for a bit to repair your respiratory epithelium, and you're fine. The flu is 100% gone. So this immune response is killing the cancer cell, and then goes and can kill every neuron that was expressing whatever protein this cancer cell decided to turn on. And then patients can very quickly develop symptoms over the course of hours or days. So I'll show you some examples of this, and you will now be inundated into the Esoterica Society of Understanding Paraneoplastic Diseases. So you can imagine, for example, if this protein was expressed in the cerebellum, what might happen if you wipe out Purkinje neurons in the cerebellum. So in fact, that's the answer to what happens in this patient. So this is a patient who we saw at Rockefeller University has a little research hospital. It's free of charge for patients to come into, and they are patients typically who have a disease nobody understands and are looking for help, and we have an institutional review board that reviews the ethics of studying patients. And in this case, we draw blood from these patients and try and understand, get a foothold on what's happening. So this is a patient who had no cancer when she presented. Then she presented with a syndrome I'm going to show you, and then subsequently was found, nobody could find a cancer in her, and we said, well, this has to be either a breast or an ovarian cancer. We went so far as to take out her ovaries, and the pathologist couldn't find anything, and we went so far as to slice very finely the entire ovary, and she had ovarian cancer. But it was just so small because it was under immune attack. So what did it look like from the brain point of view? This is cerebellar ataxia. So it's fascinating physiology, but basically she can't control sort of how to turn on or off her muscles appropriately. And this is a classical neurologic sign pointing to the cerebellum. That, by the way, is me before I had white hair from Rockefeller University. So coning down on this, this is a brain not from this patient, but from someone else who had paraneoplastic cerebellar degeneration and gave their brain to science for an autopsy. And this is a brain from a control individual. So this is stained now with the blood drawn from this patient. And in her blood, she had antibodies that stain Purkinje neurons, and that's the cell we've been talking about here. And those antibodies, when we use them to stain the brain from a patient who died with paraneoplastic cerebellar degeneration, the brain looks fine, but there's no Purkinje neurons. They're all gone. So that's like the influenza immune response. It wiped out every Purkinje neuron. So these essentially are patients with this cerebellar ataxia who are human Purkinje neuron knockouts, if you want to put it in that sort of a mouse term. And the good side of the paraneoplastic syndromes is illustrated here. It's a different patient, different paraneoplastic syndrome. But this is a man who came into the hospital who was a smoker. Another dad warning, no smoking. Okay, this is small cell lung cancer that occurs only in patients who smoke cigarettes. Not common, but when it occurs 75% of the time, it's widely metastatic by the time it's already known. So it's a nasty, nasty tumor. So this was picked up incidentally on a routine chest x-ray, and then he had a CT scan. This is a CT cut. That's the lung in black here, but there's a dark spot, so there's a mass inside his lung. So that was biopsied at the time here, and it was unfortunately small cell lung cancer. So they sent this patient home to get him in touch with a lung oncologist and plan treatment plan and see how staging and all of that. So he came back a couple months later, and for planning his treatment, his tumor had shrunk, but nothing had happened. He hadn't gotten any treatment yet. But he developed loss of all sensation of his fingers, hands, body. He couldn't feel anything. So he had a paraneoplastic syndrome concurrent with spontaneous elimination of tumor, and when he's followed again a couple months later, it was gone. He never needed any treatment, but he suffered from the paraneoplastic syndrome. So these are documentations of this process of tumor immunity that can happen in people. In fact, this was done a long time ago. Most of this in conjunction with my mentor in the hospital, Jerry Posner, probably the world's greatest clinician, who's still on the West Side poking around. We'd like to go see him and get words of advice. So this helped lay the groundwork for the current field of immuno-oncology, which is incredibly important as a treatment for cancer. So coming back to the brain then, this immune response, we could take this blood here and use it to stain the brain of normal individuals for all kinds of different paraneoplastic syndromes. So this is a paraneoplastic syndrome from a patient who has an immune response to this protein nova that I mentioned before. Here you can see it stains the ventral part, but not the dorsal part of the spinal cord, so we named it onconeuroventral antigen, twisting words around a little bit. There's beautiful staining here. Here's another patient with a different series that I'm going to talk about on the next slide, the HU antibody. And this one stains, again, the nervous system. So the brown, I should say, is staining in the nervous system. Blue is all the other tissues. And what you can see here is that these are neuron-specific, kind of fits that model. Beautiful brown and blue, black and white staining, if you will. This one stains all neurons in the central nervous system as well as the peripheral nervous system. These are the peripheral neurons of the vagus nerve and so on. So this is the NOVA instance, and again, just to review, this is a patient who presented with, had small cell lung cancer and didn't know that he had cancer until he presented, and what we believe was happening was, again, this is staining of his tumor. He had an immune response with the antibodies against the tumor, but something went wrong and he got into the brain, not really in Purkinje neurons. So we then were able to use this immune response both to study the patient and to cone down all the way to the level of the molecule. So this is what his syndrome looked like. This is a failure of inhibition. Here his eyes are having what's called saccadic movements, but he can't stop them. So he has rapid, it's called opsoclonus, rapid eye movements. And lying down, he's okay in bed, but when he gets up, he can't stop his muscles from shaking. So he has action-induced myoclonic tremor, would be the doctor's terminology for it. So he has a very specific syndrome relating to what his antibodies binding inside of his brain. So we could then use the serum from a whole series of patients like this to do molecular biology. Here's the serum. I think, is that what that is? Yeah. Okay. And so these are called Western blots, where we can look at the proteins inside the cells, what molecular weight they are, big ones, little ones, small ones. And then we can use this to do the real tools of molecular biology, which is cloning the genes that are making the proteins that were being turned on in the tumor cell. And also allowing us then to ask, why is the tumor cell turning on these proteins that are normally made in the brain? What are they doing in the brain? Maybe they're doing something special in the brain that the tumor cell wants, even though it's putting itself at risk for being seen by the immune system. So this is an underlying fascinating question. With the tools of molecular biology, having the gene in hand, we can also start to make that translational science point. So we can make an animal model, and this is an animal in which this protein that this guy has under attack is knocked out in a mouse. And you can see the mouse phenocopies, we would call it, the human, only it's much more severely affected. And it has this action-induced myoclonic tremor and severe weakness in its muscles. So the paraneoplastic diseases became something of an obsession in a healthy way for me. I learned a lot. This is Jerry Posner. This is Lisa DeAngelis, who is the head of neurology at Memorial Sloan Kettering Cancer Center, where I go and teach. And actually, Jerry and I wrote the book on paraneoplastic syndromes. It's extremely popular. I recommend you go out and buy it. I would say, you know, a couple things. It lists for 174, but it's been knocked down. You can now get it for a mere $19.95. I'm teasing. I've probably made $12 off the book sales. But the one thing I was excited about is it got 5 stars on Amazon. In retrospect, I think that may have been my mother. And I was excited about that when I looked at the rankings. It was like extremely high ranking. And then I realized like a ranking of 1 is good and this is not so good. So anyway, a little teasing is always healthy, right? Okay, so back to this syndrome, the paraneoplastic syndrome. Here's another one where, and this is actually an interview I'm going to show you of a patient who has a paraneoplastic syndrome affecting the limbic system, which is inside of the temporal lobes and is critical for memory. So remember the patient I told you about, patient HM, who had both of his temporal lobes taken out? So this patient had both of her temporal lobes on the MRI scan inflamed, and that's why they're white here on both sides. So you can kind of guess from patient HM what her neurologic syndrome might have been. We call it paraneoplastic limbic encephalopathy. So I love this video. In part, it's Jerry Posner doing the interview, the greatest clinician in the world.
Okay, do you remember what the date is today?
It's about the 10th or 11th.
Of what month?
July.
And what year?
1984, I think, 5. I'm not sure.
Let me tell you what the date is and the name of the hospital, and for you to remember for later, okay?
Okay.
You're at Memorial Sloan Kettering,
Right.
and it's Friday, February 2nd, 1989. Now, can you say that for me?
Yeah, Friday, April 2nd, 1889.
No, listen to me. Friday, February 2nd, 1989. Now, you say that.
Friday. I cannot concentrate on it. I can't. Go ahead.
Okay, so of course one of my favorite videos I'm sharing with you. She's wrong at the end. The patient says, I cannot concentrate, go ahead. So actually she's concentrating. She's concentrating big time. And Jerry Posner's finger comes up and says, listen to me, right? So that's different. If you are on drugs or have a metabolic encephalopathy from whatever reason, you can't lay down new memories, but it has nothing to do with the hippocampus. Your brain is clouded. But she's concentrating. She's paying attention to what Jerry is asking her to say the date and the place, and she can't lay down the new memory. So when I go into a patient like this's room, I can spend 20, 30, 40 minutes talking with them, doing an exam, and then walk out of the room for a minute, then come back in, and the patient has never seen me before, never. So they can't lay down any new memories. So it's an instance of patient HM. But in this case, what's different is we know what's going on with her, a little bit at least. So what is going on with her? So these two instances in particular have been really fascinating for the lab to study. We can draw the blood from the patient. We can use it to stain sections of brain, and we can use it to clone the genes that are making the proteins that are under attack. So in this case, the antibody stains neurons, and when we clone the gene, we can see that the RNA that's expressed is expressed in the brain here with a B, but not in any other tissues, and it's expressed in the ventral nervous system, as I showed you before. This patient's serum stains, again, neurons, and it stains proteins only in the brain, but 0 in other tissues. So that fits the sort of model for what's going on in the paraneoplastic syndromes. It's a brain-specific set of proteins that are being discovered, and that's why they're so immunogenic in the tumor. So the surprise then becomes, what do these things code for? What kind of protein is it? So you got to buckle your seatbelt for this, because this is a wild roller coaster, okay? These turn out to be the tip of an iceberg of something that was previously undiscovered, unknown in science, which is the brain has its own system for making proteins that regulate gene expression, but not at the DNA level. They don't bind to DNA. They bind to RNA. So the brain has its own system for regulating RNA after it's been transcribed from the DNA. Why would the brain have that? So it's an active, interesting scientific question. So to take you through some of the thinking here, here's a Purkinje neuron, your favorite now. What would and why would the nervous system have brain-specific RNA binding proteins that no other cell in the body uses? No immune cell, no liver cell, no spleen cell, et cetera. So there's a couple of conjectural ideas about it. One is within the nucleus itself, and these are expressed in the nucleus, these may regulate the process of RNA metabolism after it's been copied from the DNA. So I mentioned earlier, and you recall, RNA is copied as a long copy from the DNA, but then it's chopped into pieces. And what we term that is the ability to have these pieces are exons, and the piece that's spliced out is an intron. But there are instances like this one where the exon can either be in or skipped over, out, back to the binary computer, right? But this is now at the RNA level. So this exon can either be in or out, so it's a 1 or a 0, now at the molecular level, within a neuron. So many, many transcripts in the brain will have, say, 10 or more of these alternative in or out exons. And if they're randomly used, it doesn't have so much information. But if they're regulated by an RNA binding protein that can say, okay, in this case we're going to use it, and in case Y we're not going to use it, then it becomes an information point. And if a single gene has 10 of these, each of which can be independently regulated in or out, then you can do the math, 2 times 2, 1, 0, for each of 10 things. So you can make 2 to the 10th different proteins. And factors of 2 go exponentially very quickly. You can have ones that are 15. So if you had 10 alternative exons that can be in or out, you can make 2 to the 10th different protein variants from one gene. That's 1,024. So you can increase this potential coding capacity of the genome from 30,000 gene encoding proteins to 300,000 just by this mechanism alone. Another thing neurons might be interested in doing is, well, the DNA is stuck here in the nucleus, the RNA is not. So it comes out to be translated in the cell body, but it doesn't have to stay even near the cell body of the nucleus. It can be transported out into these dendrites. So an active area of science right now is looking at, is there RNA out here in these dendrites of the Purkinje neuron, but of every neuron in the brain? And can that localized RNA mediate little pieces of genetic plasticity, genetic encoding, differently at this dendrite and at this dendrite, in which case all of a sudden the 10,000 or 100,000 dendritic spines could be independently regulated using the biology of genomic evolution to make our brain plasticity maybe special. And then the other point to make, I got 4 points. The other point here is that we know now that there's a growing list. Paraneoplastic diseases are sort of one, but there's genetic diseases as well that I'll mention briefly, in which RNA binding proteins, regulators like this, are impacted or defective, and that can lead to neurologic disease. So we know that it gives us that sort of confidence, and another point to study this relationship between brain disease and RNA regulation. So the fourth point here is just to go back to basic science and the integration of this kind of thinking with basic science of RNA. So I just can't resist showing you one slide. This is a talk I gave in Zacatecas, Mexico in 1989, in which I disparage protein, which I really like to do at Cold Spring Harbor when I go lecture there, because that's where Jim Watson is. But Watson was a dogmatic fellow in many negative ways, and one of them was that he is the one who pushed the central dogma that you're all taught probably in high school, right? Which is that DNA is the key molecule per Watson's iteration of this. And it is true that the DNA is important and critical for transmission from generation to generation of your genetic material. Then DNA is then made into RNA, and RNA is translated into protein. RNA, for many years after the central dogma was put forth, was essentially an ignored intermediate, kind of annoying to deal with, right? But the main point was DNA is your genetic material, and then protein does the work in the cell. And what this is pointing us to in the slides I've showed you is the importance of RNA. But there's another way to look at this, which turned into a Nobel Prize not too far after the central dogma was put forth. So that came from basic science. So I just can't resist going through this a little bit with you. So this was a basic scientist in Colorado, sort of pretty much obscure laboratory, discussing an incredibly obscure organism, basic organism called tetrahymena. It's a single cell, nucleus, cytoplasm. It's got all these hairs on it. It's like nobody in the world would want to study this thing. And certainly you wouldn't think studying this would have any general importance. So the guy's name was Tom Cech, and he went on to win the Nobel Prize from the slide I'm going to show you. He was doing this work with a postdoctoral fellow named Art Zhao. So they did this experiment. They basically squeezed out the juice of the tetrahymena and added it to one of the tetrahymena pre-messenger RNAs that had an intron in it still and 2 exons here. And they were trying to see what in the juice here was necessary and sufficient to cause this to be spliced from a long pre-messenger RNA into a short spliced exonal messenger RNA. So here's the diagram, right? This thing is longer. This middle piece, the intron, is going to be cut out, and then it's going to make a shorter piece. And the assay they set up was to basically squeeze the juice out of the tetrahymena. This had already been known in other systems, but Tom decided to do it in tetrahymena. So here on this is a experimental blot. I won't go into the details, but they radioactively labeled this long piece, and here it is running long on the gel. And then after putting the juice on it, it ran shorter and was able to cut this intron out to make the mature final product. So that was great, and Tom Cech was excited about this and went back to his postdoctoral fellow, Art Zhao, and said, well, Art, you know, this is a great experiment, but you screwed up one thing. And Art's like, what? He's like, you forgot to do a negative control where you leave the juice out. And so I've illustrated that here. Here, this lane got the juice, and this one he was supposed to leave it out. But even though he left it out or said he did, it spliced anyway. So it didn't have any tetrahymena anything, and it was just the pure RNA, nothing else but synthetic pure RNA. So he's like, Art, you got to redo this. So he made Art redo it for 6 months every day. And Art got the same result every time, that when he left, here's a little pearl, by the way. When he left the juice out, it cut anyway. So that told us something so profound, and I made a special picture out of it, okay? All right, this is a Bob Darnell picture for you, okay? What this told us is something so profound, it shook our understanding of the origin of life on the planet Earth. And that's fundamentally correct to this day, which is that what we believe is the way life formed on the planet Earth was a membrane with nothing in it except a pure RNA. And pure RNA, because it's single-stranded, unlike the relatively boring double-stranded Watson-Crick double helix, RNA can fold back on itself. So this RNA could fold in loops. It's got G, C, UU. They can fold back and form a stem, and then it can have a loop and another stem, and they can form in complex structures, which starts to look a lot like a protein. Now, instead of every space being an amino acid where you have 20, it's only got 4, G, A, C, and U. But still, it can make some pretty cool shapes. And in fact, this shape acts as an enzyme to cut itself out with nothing else in there. So the RNA acts as an enzyme. It acts as a protein, essentially. And that's why, with many, many other pieces of data, led by another Nobel laureate, Jack Szostak, who's brilliant, the current thinking is that the things that happened in the primordial Earth were just the right conditions to have nucleotides that could be encapsulated inside a membrane and form chains long enough so they could act as enzymes to replicate themselves. So you have RNA replicating RNA, and then that membrane can divide into two. So it's a primordial cell. So anyway, coming back to reality, I do like that story. There's another set of syndromes that hammer home this point of thinking about clinical medicine and molecules. And this now is a genetic syndrome as opposed to paraneoplastic diseases, which are more complex. This is called the Fragile X syndrome, and this is super common. 1 in 4,000 boys who are born worldwide have this mutation. And here's the protein called FMRP or FMR1. And we were attracted to this very early on in the 1990s because when we cloned NOVA, and then FMRP was cloned by Steve Warren and others, it had the same overall architecture, a little different, but it reminded us a lot of NOVA. And we and others began to study it because maybe it too was an RNA binding protein. And I'm not showing you the data, but NOVA turns out to be an RNA binding protein, as is FMRP. So the point about FMRP here is not only is it implicated in the disease, but it is implicated in a very specific way, which is that most kids who have the Fragile X syndrome lose the ability to make any of the protein. They have a mutation that stops the gene from being transcribed from DNA into RNA. There are occasional cases of kids where the protein is still made, but there's a single point mutation. And that single point mutation we know now, right in the middle of this RNA binding domain called KH2, blocks its ability to bind to RNA. So that tells us that intellectual disability can be caused by an inability to regulate RNA. So this thing down here sits and it regulates the translation of mRNA into protein. And if you don't regulate that RNA translation properly, you become intellectually disabled. So fast forwarding from that to the theme of the talk, intellectual ability and its correlate, memory, requires new protein synthesis. We know that's true at the level of the synapse. I'll tell you about that in 1 second. And we know that's true at the level of the genetics, such that key RNA regulators also are key in intellectual function and memory. And that relates to brain-specific systems for regulating memory. So tying how all of this works together in the last couple of minutes here. So here's this picture of the synapse, the axon, and the dendrite. Here are the receptors in the dendrite that respond to information coming in. So here's an axon with all its vesicles secreting neurotransmitter if it fires onto the receptors, let's say glutamate. And briefly, the way this works is that glutamate will bind to the receptors and allow sodium ions to flux through and an electric current to pass from the axon into the dendrite. So that's the electric circuit. Now, if, going back to the first slide of the beach and the memory and coincidence detection, the way we think memory forms at a truly molecular level is that it requires coincidence detection. So if I'm dancing on the stage and you're hearing my voice and seeing my wonderful green sweater, all of those things together may help you remember this slide more. And at a molecular level, the concept is there's a second channel here that also binds glutamate, but it doesn't open up. So this channel, which is called a glutamate receptor channel, instead, when glutamate binds, you can see it has a divalent cation, magnesium, that blocks the channel. And when glutamate binds, it transiently wiggles and opens up and allows the magnesium to come loose. That makes a nanosecond window so that if a second axon fires, you get coincidence detection. So while this magnesium is out, just in the nanosecond that you're having, let's say, the smell of the ocean here, if you get another firing of an axon that has the sound of a seagull, then you get a second bolus of glutamate. And that second glutamate can bind when magnesium is gone and now allow a divalent cation to come through. Instead of potassium or sodium here, it allows calcium to come through. Calcium is a key second messenger inside cells. It's a signal molecule. It says something special just happened, do something. And we don't have time to talk about it, but that do something is really important. It's something Daniel's interested in studying, for example. So calcium entry is a signal for changing the nature of the synapse. Let's make it stronger. Let's make it more robust. In a whole bunch of molecular ways, we can start to understand by thinking about all the information we've put together here. So as it turns out, as I alluded to with that green Purkinje neuron, there are RNAs sitting out in the dendrite just waiting for some action. And those RNAs turn out to be probably activated by calcium, which then triggers local RNA binding proteins, NOVA, FMRP, ELA-V, a whole list that are also sitting out here. So there's RNA, there's regulatory proteins, and they're just triggered waiting for coincidence detection to allow calcium to come in. When all of that happens, bingo, there's a molecular mechanism for memory. Okay? So that's mostly what I wanted to say, but I'm going to go on a little bit because I just want to show you a movie of how this relates to actual clinical medicine. How can we translate this stuff? So here's a disease called spinal muscular atrophy. It's a genetic disorder, and it's a defect in this gene called SMN1. Unfortunately, it leads to infantile paralysis and death. So it's uncommon, but it's sort of an infantile form of Lou Gehrig's disease, if you will. Now, scientists, led by a man named Adrian Krainer, recognize that there's a second copy, as there often is in the genome, a duplicate copy of the SMN gene called SMN1 called SMN2, but it doesn't work so well. In fact, it doesn't really work at all because it has a splicing defect in it where it's made in a way that this exon 7 gets skipped. And it gets skipped because certain proteins in the cell force it to get skipped. And so Adrian pursued this in a way that I'm not going to go into, but it's skipped because something is inhibiting the use of exon 7. It's an RNA binding protein that comes right over here and blocks the use of exon 7. So it works because it's binding a specific sequence here and blocking the use of exon 7. So what happens if you take something that can block that inhibitor? So this is called an antisense oligonucleotide, and it blocks the inhibitor from binding there. All of a sudden, you go from a case where exon 7 is skipped to a case where exon 7 is included. So that was done in the test tube, experimentally, then done in mice, and then they went ahead and made a drug called Spinraza. This is the first FDA-approved drug ever, not only for this disease, but the first FDA-approved drug that was an RNA antisense molecule here. We can talk more about this. But it turns out it works in vivo, that the SMN2 protein is restored. So even though the kids who have this disease don't have any SMN1, that's not coming back. We can turn back on and rescue the loss of SMN1 by rescuing this splicing problem by this antisense red oligonucleotide. So does this work? Again, these children are grievously wounded by their genetics, and they go on to die over the course of weeks. But this is a movie of real life here. This is made by the parents. You can see him breathing, barely able to breathe. Can't hold his head up, and his arms are floppy.
Cameron is now 7 weeks old.
Now he's been given this drug.
Yay, look at you. Yeah, good boy.
He's way beyond his expected life.
You got it. You did it. Oh, so proud of you, little man.
See, I'm a dad.
Push, push.
4 years.
Push, push, push. Push, push, push.
Yeah, keep going. Push, push, push. He is riding a freaking bicycle. So take that, SMA.
Right.
Oh my God.
I mean, yeah. And I got 4 kids, and I can't watch this without getting a little teary. So this is the full circle of observing clinical medicine, understanding it down to the level of the molecule of, in this case, RNA regulation, and figuring out how to rescue that and rescue this child. So I'll close by just saying what future directions might be around for you guys to think about. RNA is important in all aspects of memory, all aspects of cognitive function. We have a big problem in a disease in the world of Alzheimer's disease, which is a memory failure. And some interesting stuff has come out to think about. This is not RNA now, but you can put little electrical stimulators deep in the brain in the area that's important for memory formation. This is near the nucleus basalis of the basal forebrain, cholinergic forebrain. Stimulate that and then assay immediately thereafter how an Alzheimer's patient does. So this is an Alzheimer's patient asked to copy these figures and just can't really do it, right? Then this electrical stimulator is turned on and they do a lot better. So this is not super, super, super in rock solid science at this point because these are at the frontiers, literally, of experiments that are being done. So this is quite fascinating to think about. So the technology is moving very quickly is the point of this slide. And what one would like to do would be to think about more refined ways of treatment that could actually last and be forthcoming, which would require understanding the molecular basis of what's going wrong in these patients. So this is my last slide, I believe. I think, you know, what I've tried to convey to you is that human disease in the brain in particular, but lots of human disease requires us to go from the clinic to the molecule and back again. So it's a long journey. In the middle of that journey includes RNA for many diseases. This is my own thinking, and my lab knows that's coffee stain here, but of how I think this might be working. And it's more complicated than I understand, so I'm going to depend on you guys to figure it out. But the bottom line is it's a fascinating time to be thinking about doing science and relating it to people who are in need. So, you know, as you're aware, science is a team sport, like hockey, soccer, basketball, life. And being a team sport, you're happily dependent on a large group of people. This is the current group in my laboratory. You know, long list of collaborators I've had, support that we've been given by funding agencies, and a long list of key collaborators. I have a lab barbecue every year, as Daniel knows, and you're all invited. And then this is my current laboratory. It's just a lovely group of thoughtful, careful, selected people who love science. And then these are my collaborators. That's my daddy, who's also at Rockefeller, 92 years old, going strong. That's Jerry Posner, my clinical mentor. That's my thesis advisor. That's my other thesis advisor, Bob Rader, Nat Heinz, and that's me. So, basta. Thank you so much for your attention.
Maybe some 3 questions. I'll take 3 questions. All right. So now we have some time for Q&A, and as promised, we have some gifts for those of the brave souls who are willing to ask their questions. So just wait for the mic to come, and it looks like we have one already going. So why don't you go ahead and ask your first question?
Can you say your name and where you're from?
Yes, please say your name and where you're from.
Dr. Darnell, my name is Matthew Junko from Newcomers High School. My grandfather got coronavirus and suffered a liver disease. When asked what year it was, he said 1991. What explanation do you give for that?
for
Okay.
liver disease in 1991. Sorry, can you repeat the question?
He got coronavirus, he had a liver disease as a background, and when he got to the hospital, they asked him what year it was. He said 1991.
I see, got you. So he was disoriented. Yeah, I like questions like this. I'm a neurologist, I can answer anything. And nobody can challenge me because there's no other neurologists in the room. But your grandfather, I'm not sure how old he was, but it almost doesn't matter to me. It sounds like when he was sick, he was confused and got the year wrong. And so that would a little bit in my mind fit what I was alluding to when the woman was asked, what year is this? And one of the things we exclude in terms of was she having a limbic encephalopathy with temporal lobe dysfunction or dementia or something like that, is we exclude metabolic things. So when you're sick, you make all these cytokines, they make you ache, they make you feel terrible. Your brain doesn't work so well either. So especially on a baseline where you're elderly and maybe other things are going wrong with your body, is very understandable and I would guess very reversible. Okay?
Okay, so.
Yeah. I'll send a bill for that consultation. Yes.
Okay, we have a question.
Hi, I'm Elizabeth from Columbia Grammar. In the beginning, you were showing images of like neurons singled out in the brain. How exactly do you get them if there's so many of the neurons? Is it more like radioactive tagging?
How did they get pictures the way I was
Yeah.
showing? It's done with genetics. So they put in molecules that color a neuron, and there's different barcodes for different colors, and they put them all in a mouse brain. It's the same mouse brain, but different colored tags can be put in that way. And then they can image red, and they'll show that neuron. They can image blue, and they'll show that neuron. And they can, the technology is beautiful. It's Genelia Farms, so maybe genelia.org. I'm not sure the website is there, but you can look it up and you can play with it. It's publicly available.
So, hi, my name is Abdullah Abuzaid. I'm from Newcomers High School. So my question is, what is the reason behind not remembering a lot of memories I had in my life until it be triggered by an objective that was part of the memory itself?
So you're asking about long-term memories and how they were triggered?
Yeah, because like I don't remember these memories until like someone told me something about it, and then I remember everything. So that
Right.
sounds weird.
Good. No, that's right on point. And I think we don't really understand it in detail, so I can't give you an absolute answer, but our model in terms of the molecules has to do with what I was showing at the very end slide, where a coincidence of my move, or let's go to the beach, a coincidence of the seagull, the sound, all these sights, sounds, and smells. In particular, smell is a big input for memory for us and for animals, right? They know where to go pee. Again, they use that as a territorial marking. So those coincidences come together for animals and has stayed with us as humans to nail that memory down. Like there's a book called Why I Love My Daddy that I used to read to my kids. I don't know if any of you guys have ever had that read to you. I highly recommend it. So it's a cute little picture book along these lines, and one of the pictures I love in there is there's a fox with 3 little baby foxes next to it. And there's a porcupine there. And the fox is telling its babies, don't mess with that guy. He may look tasty, but he's going to hurt you. And the reason, we don't know how that kind of memory gets taught, and it may not work so well. So one of the little porcupines a few days later may go out and try that, say, I don't know if my dad was right, and get poked by that thing. And then he'll have forever seared in his brain the coincidence of the image of that porcupine, the smell of that porcupine, and the pain of that porcupine. So that coincidence will trigger something in the limbic memory part of his brain that will enable 2 axons, 3 axons, 4 axons to fire in one spot where was that porcupine, what did it look like, what did it smell like, and what did it feel like to touch it. And that will release a massive amount of calcium on not just one, but an array of dendrites. And they will be fixed and forever changed. The model is that one of the things they will do is affect the RNA regulation in those array of dendrites, such that they, for example, will now make more glutamate receptors. So at the same stimulus, seeing a porcupine is enough, or seeing and smelling one is enough to re-stimulate that pathway because there's more glutamate receptors now. So the same input will like massively stimulate that little porcupine for like, okay, I'm not doing that twice. Okay, next question.
Hi, I'm Avi Zimmerman. I'm from Nightingale. My question is about the Alzheimer's slide that you were talking about. Is it dysfunction of the synaptic connections, or is it degeneration of the neurons entirely? Because I was wondering about the magnetic stimulation and how that works.
Right, okay, lovely thought. So what we believe is it's some combination of the two. It's not like the picture I showed of the paraneoplastic cerebellar degeneration, where every neuron is gone. That patient has irreversible loss of cerebellar function. In Alzheimer's disease, we believe the plaques and tangles, which are at the root of the disease, cause dysfunction, death, and little dysfunction on a array of different neurons. So while there is loss of neurons in that basal forebrain, for example, which is the earliest area affected in Alzheimer's, it's not a complete wipeout. So the idea is it's not working so well because the pathway is defective by virtue of dysfunction, loss, and some combination of the two for that group of neurons. But if you stimulate it acutely, you can transiently reactivate that pathway with whatever is left there, and they can then draw, in the acute setting, a better copy of that test. So it's a combination of some loss, some dysfunction, and some normal neurons still there. So it's encouraging in that sense that if you could identify patients early enough, you could intervene perhaps by transcranial magnetic stimulation, which is what that was, and perhaps by drugs that are rescuing parts of the pathway that's defective or could be enhanced, like the electrical.
So we should probably, because we are running a little over, there's plenty of time to ask Bob questions in the Ask Bob Everything section. But I just want to create a few announcements for everybody for some wayfinding. But before we do, let's give Bob an amazing round of applause.
Thank you.